Right, we are learning a light-dependent reaction. Now, light-dependent reaction, first we have to draw the complete structure of the thylakoid membrane. Thylakoid membrane, we have a photosystem 2 and photosystem 1. And the rest of the proteins complexes are important. And here you can see 3 complexes and again 3 complexes. The name of these 3 complexes are not required to remember at this level. They have a plastoquinone, cytochrome and plastocyanine, and these are ferredoxine and ferrous sulfur and NADPH dehydrogenase. These are the names of these proteins, but at the moment we don't want these. So just keep the names somewhere else and then we try to learn how it happened, right? And the last thing I just want to add, again as similar to the electron transport chain and oxidative phosphorylation, we can also find ATP synthase complex. So ATP synthase complex we can find here like this. I'm drawing the ATP synthase complex. It is also there. So this is also a little bit of interest in that electron transport chain and light dependent reaction. They have some similarity. Probably it provides the evolutionary. They had starting point is same, but once they change, one become chloroplast, the other become mitochondria. Right, let's start the process now. Again, just to remind, this photosystem has pigments, chlorophyll, and then accessory pigments and proteins. Right, the process starts at the PS2 and it absorbs the sunlight. When it absorbs energy of the sunlight, all these molecules, proteins, pigments, carefully take this energy of the sunlight to the reaction center. Now the reaction center is loaded with the energy of the sunlight. So this sometimes remember us when you do a small experiment taking the magnifying glass. We holding magnifying glass to the bright sunlight, bright sunlight, and we can focus the sunlight into one tiny bright spot. At this bright spot, we can actually burn a paper or something. So magnifying glass can absorb this. It's not absorbing, actually. It's focus all the scattered light into one focus center, and then the light energy is concentrated. So we can convert into heat energy. It's a little bit similar to that kind of a thing. The energy of sunlight focused to the reaction center. Then the reaction center has this chlorophyll, and the chlorophyll is made out of carbon, hydrogen, oxygen, nitrogen, like atoms, and also magnesium. Now what happens, this energy is actually transferred to these atoms which made these chlorophyll molecules. And we can easily understand if finally the energy is transferred to the atoms, who is getting this energy? Electrons. Because electrons can absorb the energy, and the electron is moving to the next level. That means if it has the different shells, they are moving to the different shells. But the valence electrons, that means the outer shells electrons, when they absorb the energy, only option is they have to escape from the atom. So as a result, outcome is electrons are released. Now I am showing only one electron, but it's not a one electron, it's a clouds of electrons released from the photosystems. Now we have a two option. One is what happened to the atoms which lost the electron. Second one, what happened to these free electrons with energy? Right? So I make a red circle around it, showing that loaded with energy. Both conditions are not stable. Because atoms lost electron, they are unstable. These atoms are highly unstable. And the electrons with energy is also unstable. Alright, now the electron has one option. That means electron can move towards these electron attractive protein complexes. So electron can move through these protein complexes just like a electron transport chain. Now the photosystem 2 is unstable as they lost the electrons. As a result of that, some of the components of the photosystem, which is not much discovered yet, what is exactly happening inside the photosystem 2 when electron left, but the final outcome is clear. Final outcome is components of the photosystem can breaks down a water molecule into a oxygen. So they can break down into the oxygen, and some of the textbook they first break down this to hydroxyl group and hydrogen, and then this hydroxyl group again they try to form water oxygen molecule. But I think Cambridge books use this reaction. So if you want to balance this equation, it's half. We can take here half. Then we can solve one problem. This electron can move back to the photosystem. So this electron coming from the water molecule again stabilize the atoms inside the photosystem too. So they can give back this electron and the atoms are stable in the photosystem too. Okay, we solve one problem now. Again atoms are back to normal state, they are stable. Right, now what happened to the energy of this electron? Now energy of the electrons this time is slightly different than electron transport chain. So it pumps hydrogen from outside to in. The outside is a stroma, and there's a thylakoid. Inside the thylakoid is this area. So hydrogen ion pump into the thylakoid using this energy of the electrons. Right, now this reaction is called photolysis. This is called photolysis, splitting of water molecule. Now we understand in the photosynthesis, oxygen molecules are formed as a result of splitting of water molecule. That means photolysis. Okay. Still, we have a problem with this electron. It lost the energy, but electron is free now. So what is going to happen to this electron? So we start now photosystem 1. Again, story is same. Light energy is absorbed by the photosystem 1, and the energy move into the reaction center, and the electrons are emitted, excited out of this photosystem 1 with the energy. Okay, now again two issues left. One is photosystem 1 atoms are not stable because they lost the electron. Second one, the free electrons with energy is not stable. Here there is no splitting of water. Then these electrons can move into the photosystem 2. Electrons coming from photosystem 2 move into photosystem 1. These electrons can provide the electrons to atoms which lost the electron from photosystem 1. Then electron is going to the atoms of photosystem 1 to become stabilized. Then the photosystem 1 gets stable. Right, then next round these electrons again moving through these complexes. And in this case, again different, it doesn't use this energy to pump hydrogen ion in. So photosystem 1 electrons do not use its energy to pump hydrogen ion in. Instead, this electron energy preserved, and it's going to add to another molecule. So in this case, the electron acceptor molecule is NADP, nicotinamide adenine dinucleotide phosphate. So it can combine with these electrons with the protons to make a NADPH. Now the energy is transferred to the NADPH molecule. Energy is not used by these electrons. So with the intact energy, it moves to the NADP molecules to make NADPH. So NADP is oxidized, NADPH is reduced form. So then NADPH is formed. So far what are the products? Oxygen, and this is product A, and this is product B is NADPH. Right, we just again label these issues, the events. Number 1, light is absorbed by the photosystem 2. Number 2, this energy used to emit electrons. Then this electron with this energy is moving through these protein complexes or electron carrier proteins. And that energy is used to pump hydrogen ion in. And to become stable, this photosystem 2, the photolysis take place. That is, water molecules split into hydrogen ion, electron and oxygen. And this electron is moving back to the photosystem 2. Then the event 7 is light energy is absorbed by photosystem 1 and the electron is excited, and these electrons are moving towards these complexes. But the energy is not used to pump hydrogen, instead energy transfer to NADPH molecule. Once this electron left from photosystem 1, the electron is coming from photosystem 2 is going to photosystem 2 at the 11th step. Right now, last thing, what is going to happen, the hydrogen ion start to build up inside this thylakoid. When hydrogen ion build up inside hydrogen ion is increasing. Stroma hydrogen ion is decreasing, which is also change the pH, because inside pH is getting lower now because more protons inside the thylakoid. It's again same way like in electron transport chain. One side has low hydrogen ion, other side has higher hydrogen ion. As a result of that, it form electrochemical gradients. So electrochemical gradient establish a potential difference. So one side energy is high, other side energy is low. Then only escape route is this hydrogen ion has to go out of this ATP synthesis complex. So when hydrogen ion is moving through this ATP synthase, this energy is used to produce a ADP molecule combined with phosphate to make ATP molecule. That's a C molecule. ATP is formed. It's similar to the electron transport chain and oxidative phosphorylation, but this time energy is not the glucose, the energy is sunlight. So using sunlight energy, production of ATP is called photophosphorylation. And you can see this all the reaction is ending at one end. So it is starting from here and ending at this place. So it's a non-cyclic. Now you understand what is non-cyclic photophosphorylation. So completing the event, number 12 is, that is a chemiosmosis again. Chemiosmosis is one side has a higher hydrogen ion, other side has lower hydrogen ion, which make a potential difference. As a result, high potential to low potentials, hydrogen ion moving through this ATP synthesis complex is called chemiosmosis. That energy is used to make ATP, that is a non-cyclic photophosphorylation. Right. On the notes, if you see there are 17 points listed one by one. And here again we have at least 13 events making this one. So which is very close to the points of these nodes. Each point is listed very clearly on the node section. So when you if you understand this process and go through these points, and which explain all the events of this diagram. So best when you study this light dependent reaction, you have the diagram, and looking at the diagram, try to list these event by numbers. If you can't list one event, you can check the notes. Starting with one, okay, light is absorbed by photosystem 2. Then the energy used to emit the electrons. Electrons are excited and then released from the atoms, that is 2. 3 is this electron is moving toward the thylakoid membrane proteins, and this energy is used to pump hydrogen ion in, that's the event 4. So to study this light dependent reaction, the best way is once you copy this down, try to list each event until you get remember these events. And if you have a difficulty what is the exact process of this event, check the points in the notes, then you can find this event. Right, any questions about this non-cyclic photophosphorylation light dependent reaction? Did you copy this one?
Yes.
There's a blank page which is actually a listed non-cyclic I mean it's a A4 sheet. Usually best maybe you can start from this orientation, landscape, portrait and landscape orientation. You have a more width. I think we can fit it, but anyway, if you can manage even the portrait orientation, the length is enough. Then we finished the light dependent reaction in one page. Okay, just moving forward, what are the products of light dependent reaction? There are 3 products. Oxygen is released. The plant produce oxygen by photosynthesis by splitting the water molecule, and oxygen released. And the other 2 products are NADPH and ATP molecules. So B and C are the most important molecules produced that they can use by the plants, because oxygen is not used by the plant. In this case, oxygen is released, and this happens during the daytime. Then plant can still use ATP, because if it is produced ATP here during the daytime, they can use this ATP, and also they can convert this ATP into other molecules for the storage. Okay, we are moving to the next process, cyclic photophosphorylation. Cyclic photophosphorylation is not that important, because mostly non-cyclic photophosphorylation takes place in the plants. But cyclic photophosphorylation is an event which takes place during special conditions, and maybe a photosynthesis organism. They use cyclic photophosphorylation when the conditions are not favorable. They can still produce some amount of energy. Okay, let's see then cyclic photophosphorylation. Well, I'm going to add same thing again very quickly. Photosystem 1, photosystem 2, first one, then the photosystem 1, and we can also find electron carrier proteins, and then ATP synthesis complex. Yes, which is similar to the previous diagram. We have photosystem 2, photosystem 1, electron carrier proteins, and ATP synthesis complex. Now in cyclic photophosphorylation, only the photosystem 1 is active. So photosystem 1 is active. As a result, light is absorbed by the photosystem 1. As the light energy is moving to the reaction center, this energy is used to emit electrons, which is same like non-cyclic one. Then the electrons are released. Now this electron has energy. Now this electron is moving to the electron carrier proteins like this. So it's moving to this first one, and after that the difference is it is moving to the other electron carrier proteins of the photosystem 2 like this. You see the difference, it's just moving to the first electron carrier proteins, then it's going to the electron carrier proteins of photosystem 2. Now photosystem 1 atoms are unstable because they release electrons. Now this electron is coming back to the same place. It solved the problem. Electron is going back to the atoms which lost electrons. Okay, now you can see this is a cycle. We can see a clear cycle in this case because the electron which is coming from this is going back again to the same place. But when electron comes to the atom, it lost the energy. Right, what happened to this energy? It used to pump hydrogen ion in. Now same way the protons or hydrogen ions accumulate. It's now very similar to the previous event. Then it creates one side less ions, other side higher concentration of ions. When the ion is partitioned by a membrane, it creates electrochemical gradients and it establishes a potential difference. So one side potential is high, other side potential is less. Then this hydrogen ion can only escape through this ATP synthesis complex. At this point, the energy now transfer back to this ADP molecules to phosphate group, and then it makes the ATP molecule. I mean again the photophosphorylation. So here only the difference is only ATP is formed, and what is not formed, oxygen is not formed, and NADPH is not formed. It's not formed. Oxygen not formed, NADPH not formed. It just to produce ATP. I think there is no very clear, good explanation when this is happening, why this is happening, and some document they say if the plant is running out of water, when they have a less amount of water, they can conserve water, because if the photosystem to operate, it split the water molecules. So water molecule is decreasing, and they can conserve water, but still they can produce energy by producing this ATP. So that kind of a situation, they can produce a cyclic photophosphorylation pathway. So it just produces only ATP. Its significance is not very great, but mostly the photosystem non-cyclic one is the predominant photophosphorylation of plants. Alright, so this is the most important part of the photosynthesis, because a lot of questions based on the non-cyclic photophosphorylation. And in this chapter, the most important lesson is the light dependent reaction, and the next one coming up later, that is light independent reaction. So together, light dependent and light independent reaction is the core of this photosynthesis. That means if the student think it's too much to remember this light dependent, light independent, all these reactions, but you don't have many other things to remember. It's only the light dependent and light independent reaction. And you take the energy respiration, glycolysis, link reaction, Krebs cycle, and electron transport chain. That is the important part of the chapter 12. So chapter 13, light dependent and light independent reaction. Okay, so now we have to move to the next level, because we learn the photosynthesis in the lower level classes like this: carbon dioxide, water, it produces glucose molecule and oxygen molecule. And so far we have learned, yes, oxygen is produced, because here non-cyclic photophosphorylation, oxygen is produced. And we also see importance of water molecules. Photolysis, water molecule is important. But we haven't seen where is carbon dioxide, where is glucose. Now when we come into the next lesson, which is light independent reaction, that means it doesn't need light. Right? It doesn't need light. You can understand the energy produced during the light dependent reaction. So ATP plus NADPH need for this reaction to fix carbon dioxide with the water. It need energy. But here when we see the reaction is water and carbon dioxide joining, actually that is not the realistic situation. You will see the water molecule is important to photolysis. but not fixing water into the carbon dioxide. But when we write simplified reaction, we just add carbon dioxide and water. But water is involved uh, directly in the light dependent reaction. Okay, then we try to learn next topic, light independent reaction. So we could not finish maybe today light independent reaction, but we can continue next time light independent reaction. This is also called a Calvin cycle. But the person who actually discovered this light independent reaction, his name is Calvin, and to make it honor to this person, we give the name Calvin cycle. Now, where does Calvin cycle take place? It is take place in the stroma. It's not take place in the thylakoid membrane or thylakoid inside. It is happening in the stroma of the chloroplast. Okay, it is a cyclic process. It is cyclic process, and we can find three important molecule in this cycle. First one, we're going to see the name soon, and the second one, and the third one. And there's an intermediate molecule is here. And what is going to feed into the cycle is carbon dioxide. So carbon dioxide is joining. So if you remember Krebs cycle, acetyl-CoA is joining to the cycle. And photosynthesis, carbon dioxide is coming into the cycle. Right, now let's see the first reaction. So first reaction, carbon dioxide plus ribulose bisphosphate. Ribulose 1,5-bis, abbreviated by RUBP. So this one is RUBP, ribulose bisphosphate, combined with carbon dioxide to make the intermediate product, and then it moves to the other product. Right, I think we're going to stop at this level. We can continue rest of the light independent reaction next time. So you can add this note. I mean, anyway, I'm going to start from the beginning again, showing this cycle, and then explaining the light independent reaction, right? Okay, then see you next time.
See you soon.
Okay, bye.
Thank you. Bye, sir.